On Tuesday we talked about 3D comparisons. So today we go into sequence comparisons. Don't forget that science is communication is something that intrinsically you cannot only do in your little cell with your big computer or small computer or whatever, being all by yourself. You need, as soon as you can, it takes a while to sort of get your teeth into the program. You have to write the program, you get the teeth into the data, but then very soon you have to find people to discuss with. Otherwise, science does not happen without perspective change, or mostly not happen without perspective changes. And questions to talks are often the first step. Why do we compare 3D structures and not function? Because it's easier to compare 3D structures. The protein comparisons in principle is not a metric, because protein A is similar to protein B is similar to protein C. Does not mean that C and A is similar. Ultimately, this relates to the issue of domains. And to get this sequence comparison and structure comparison into a transitivity relation, into, here's the same thing on a structure, into something where in fact have a metric, is what Manfred Zippel contributed through COPS, and that allows a generic automated objective classification of all proteins. There's a classification of proteins into 3D, the place of the residue, the atom in a three-dimensional space, into 2D contexts or distances. This step from 3D to 1D to 2D is a reduction of 1, but it doesn't really reduce the information. Most of the information is still there, except for you cannot distinguish between the image and its mirror image. But when you go one more dimension from 2D to 1D, you lose all the contacts, connectivity, and by that you lose a lot of the important 3D information. So we begin today with pairwise comparisons. And let's just look at the first. For today I really need a pointer. I can also move it closer to, or I can just move there, right? So this is one way in which you can align it. Then you get another word. There's another way in which you align it. And then you have a bunch of simple alignments. And let's begin with the first one that most of you will know the meaning of. This is father, and then when you go a little bit further down the road, you see that, well, this is something else, and then you see that these three in three different languages are grandfather. Okay, and you can clearly somehow see that, except for the something else, they somehow have a relation. In terms of their meaning, they have a relation, they look similar. But now, actually, we have two different ways of aligning, of arranging our alignments. One way of arranging it is this way, which is the way that sort of is dedicated, is essentially given by what is most similar in some sense. This way is given by the understanding of the word. Now, what's very important here, when we talk about alignment, there's this notion of the optimal alignment, and the notion of the optimal alignment is always given some criterion that is definable. Typically, meaning is not definable. So for most of the course of this lecture, the reason for aligning proteins will be to understand that they have similar structure. So what sequence similarity do I need in order to establish that two proteins have a similar structure? Is typically the answer that we are going after. We can put that into a metric or into numbers, true, but this is typically not what people call an objective function. Typically when they talk about an objective function in sequence alignments, but it's not cold, is it? Okay, there's not much I can do. The heating is on. So what they typically talk about is something that measures, for instance, pairwise sequence identity. So optimal alignment is defined as in where I have a function, the objective function, which is optimize the number of identically matched residues, amino acids, words, letters. That then hopefully translates to a similar meaning, so structure, for instance. That again is what I said, right is defined. by the function. The correct, the simplest is percentage of sequence identity. There are more complicated functions. We will briefly talk about that. And it clearly ignores the meaning, the biological background. It ignores that we need to match structure, match function, or evolutionary relations. It's just sequence match. Let's look at the sequence, the string aligning problems. So we compare these two sequences. We can do it on in a global alignment, in which we essentially align these two, demanding that they align begin to end. So the first letter somehow aligns, that all letters somehow find equivalent letters in the alignment. All letters are used in the alignment, versus a local best match, where you in fact allow that the part of the sequence, the O doesn't have an equivalent and the I doesn't have an equivalent, the P here, so there are things that you leave out. The historical adaptation is evolutionary relation. We typically imply similarity in structure and function. Do you know what homology? Actually, let's get into the meaning of homology, and let's begin with a different word, phylogeny. So philo, race, tribe, kind, genesis, birth, or origin. You know what this is? Well, it's something that you can't recognize. That is a fair description of what I showed you. But doesn't anybody recognize what it could be? I'm sorry for the quality. Okay, let me blow it up. Oh no, when I blow it up, I already give the answer. But sort of this is the best I could do, and it's arguably not much better. This is from Darwin's 1859 Origin of Species, and those are all the figures in that book. It's a heavy book, big, really, really big book, and that's all the graphic material you will find in the entire book. So some things changed, right? I personally feel it's good that this has changed. But that is really the idea of Darwin, that we are originating from a common ancestor. The common ancestor, so here's a more modern tree in which you look at the tree of life, and some of you may have seen that a few weeks ago there was the new tree of life published that just brings home the message of how much new species are out there that we have never seen before, how much the tree of life is governed by sequencing and not by organisms that we really can cultivate. This is completely ignored in this one here. It's only to show that there is a way of looking at the tree of life that includes sort of most of life's form. You can do it in a slightly different way of drawing it. You put it on the circle. Again, you still have the tree underneath here. And then in this tree, now here is a little bit better way of looking at it. In this tree, one of the issues, we have two different trees here, and one of the main issues that people discuss a lot is what's the origin for these three here? What was first? Archaea or bacteria? And there are different answers, and there's a lot of scientific work going on into arguing why this or why that, or why anything else. It's a big word. Now, let's go into this idea of speciation. So you have two species, you have, in fact, you have a bunch of species, and then after a bunch of species, you somehow create a new species. Typically, the story is not as simple as this one here, because what typically, so essentially, at the time of Darwin, this is the way they visualized it. You have two species, they come together, and there's a new species. So there's one important issue that people today, in which today people, they still draw it that way, but they imagine it in a different way. Do you know what the issue, what I mean?
The fluidity of the species.
That is certainly, I'll get to that in a minute. That is certainly true, but it's not what I meant. What I mean, sorry, maybe my way of asking is not quite right. So we typically assume that we have two mixtures of species. So we have essentially two groups. And then we get another group, an outgroup, which again is a mixture. So we're not having Adam and Eve. We have families. And that way of thinking about evolution is intrinsically new. We get back to that again. Homology then is defined, it's a Greek word, homo logos, agreeing, same logos word. And it comes from Richard Owen, and predating Darwin here. in 43, so the idea of Darwin somehow is not really established with that book. That there is some relation is there before, it's just the way he explains it. And ultimately the relation is not the novelty of Darwin, but the idea that there's adaptation and that there is a mechanism for selection that realizes what we see. But already Richard Owen had this idea that when we look at different organisms, we see organs that are corresponding. So the arms in a whale look different than the arms in us. So there's a homologous organ in the whale, and that originally is the meaning of homology. Same organ in a different animal. So we talk, so therefore homology implies that we in fact have an idea about evolutionary relation, and it actually is defined on the level of the organ. Now, that was a few years ago. Today people use homology on the level of genes. And that in fact is a total misnomer. Because on the level of genes, genes don't, there are no two genes or no groups of genes are father and mother of other genes. As simple as that, right? It breaks the whole concept. What we actually mean is somehow to imply an evolutionary relation. So people very often use this term to imply there is a relation of the carrying organism. And then there are two different types of level of detail, the ortholog and the paralog, where the ortholog essentially is the same protein in a different organism that is essentially copied and does the same thing, same structure, same function, same role, while the paralog is duplicated after the speciation event. This is a jumping gene. So you have already a copy of that gene, you get another copy, you can do something else with it. Okay, that's the idea behind that. Now, getting back to this idea of how speciation happens. So you begin with this happy universe where all the flies are doing their thing and they absolutely happily mate and go about their daily life. And then at some point, you know, some love to eat this banana, such as I, and then at some point, you know, big disaster strikes. So the disaster is that suddenly there's water and that separates out two groups, and in fact it turns out that one group happens to find in their new natural environment more often a coconut? No, it's not a coconut. I don't believe it's a it could be a cocoa, but I don't believe it is. Really? It looks like it. It's true. So some like to eat chocolate without sugar. No, that cannot be what it means.
It looks
huh?
like it
Could be a papaya or something.
Papaya.
Yes, they're mostly papaya is. I'm happy with the papaya. It's much more the shape than a coconut or melon. Let's call it melon because they are sweet. They're equally sweet. So some like the melon sweet and some like the banana sweet. And they begin to really specialize on melon and banana, and you know, then after some time they're all happy again because they're rejoined, these two groups. But lo and behold, a speciation event has happened. What does it imply, speciation? You know.
Yes, because you already showed the slides.
Yeah.
But there are special many definitions of speciation. Right?
No.
One guy says two organisms are different if they cannot mate with each other, but then some species actually mate with each other.
That is not correct. That is not the correct definition. There's a very clear biological definition. So the first part, so he is on the right idea, but he used the wrong words.
They cannot produce fertile offspring?
Exactly. Mating is fine. But, and again, so they may all even have offspring, but the offspring will not mate again. So
some species actually mate with each other and actually produce children, right?
So then biology is a little bit more complicated, is what you say. This is true, such as?
I think human at the time, right? I mean, human was...
Human. Does anybody in this room who has not been with me last semester? Now we can only look at a subset of the people, because the ones who have been here last week, this is too trivial. What happened to human? everybody in this room is an example. But this biological statement that the offspring of two different species is not fertile, and that's the way you define essentially a new species. And we are all, everybody in this room is a counterexample. What do I mean by that?
I don't know, if I didn't mistake human mated with Neanderthals, and then basically they create another kind of, I don't know what kind of species.
That is the Homo fortalensis or something, or fortunatus is what the F stands for. So this is the happy human being, exactly. And they are all living on Hawaii. No, guys, wake up! There is no new species. We all carry human genes. Yeah. We also all carry Neanderthal genes. So 2%, roughly 2% of the genome of almost everybody in this room of 2.5 is Neanderthalensis, yes?
But the donkey and the horse
Yeah,
can get children, and in some more times the children can get children too. If there's a kind of mistake in the...
I thought they cannot. The mule, I believe the definition is, and that is exactly the perfect example, that mules cannot. Maybe with modern genetic engineering, the mule suddenly can get children. But in principle, for me, that was always the example, the case in point of the biological definition. I didn't know that, so this is news to me. But anyway, so we all carry Neanderthal genes, and which means that there was a mating event, and that we are, our ancestors were offspring of this relation. So that's a clear counterexample. But again, mostly biology believes in this model. The problem is then in detail, maybe sometimes you have things where this is where you don't quite really see whether you have different species or not. Here are some examples. But ultimately, this remains the case. And ultimately, what we have to remember is homology is a completely misleading term for whatever we do. That does not mean you will not find that. Most likely, you will find the word homology, homologous structure, much more often than the word similar structure, although this is totally nonsense. But it's just important to remember that. There's a typical example where I believe you find in the internet or in publications, you find more often mistakes than the correct use of things. But typo reminds me, clearly what is important is that it reminds us of the evolutionary connection that we assume is underneath existing. Okay, let's get back to the comparison of 1D sequences to find 3D similarities, and we wanted 3D similarities to find functional similarities. The simplest way to align is brute force. Visually, I really need my advancer here. Visually, by simply moving the sequence through and trying through all possibilities. This is not quite how dynamic programming works, because this is too high on complexity. So if you did two proteins of length L and you tried all possible combinations, then that number is too large, preventive. But what you can do is, and that is exactly what Smith, Needleman, Wunsch introduced, what you can do is map it to a problem, dynamic programming, to a subset of the problems. And the simplest idea here is you have two proteins aligned in both directions. You begin, and let's the objective function be identity of a letter. The simplest one is you start initializing the matrix by putting all the ones where things match. And then you find a simple path through that matrix. And while you go, you sort of sum up, and then at the end, you essentially know that here the score is highest, and that in fact you reach that highest score already at this point. This is without gaps. When we allow gaps, strictly speaking, without gaps we can prove that the algorithm finds the optimal solution, given the objective function. With gaps, the story gets slightly more complicated. We can still prove it under some additional assumptions, but in principle it's still doable. It's still Needleman-Wunsch. So now you do not have to move on the diagonal completely. You can move a certain number of steps in that direction or in that direction. But matching states are still on the diagonal, and that continues to restrict your space, and that continues to be the reason why this is still not exploding the ways of doing it, because you're essentially still on a very rigid structure, so to speak, for moving in that space. When you dot plots are ways of comparing two different sequences, and whenever you have a match, you have a dot. What you would see here is, so you have regions with higher density. So you have local regions of density, and then you have some patterns. Does anybody have an idea what the patterns are that you see? This is self-alignment in this particular case, so you take the same protein against itself. When you see that, what would you immediately sort of suspect? Yeah, exactly. So you would have the suspicion that what you see are elements that are repeated. So you find the same thing in the same sequence again at different points, right? And this is one way of finding local repetitions. Now, the gap issue, one way of doing the gap issue, so the score of the dynamic programming essentially is the sum of a delta functions in the simplest form, delta ij, where the delta is 1 if the two letters are identical, otherwise it's 0, minus the gap penalty times the number of gaps you have, right? And this function gives the same value for these two. Which one do you believe is more likely to be observed?
The left one.
Why?
Because the right one seems too fragmented.
And you don't like fragments?
No, so if you have a structure...
Is that your argument? Oh, wait, okay, what do you mean?
Makes me unhappy because
No. I mean, in many situations, Jade, makes me unhappy is good enough for me. When you say it makes me unhappy, I don't force you. But in this particular case, makes me unhappy is not enough for a scientific argument. So, what do you mean? No, I'd like to push Jade on this one.
Why?
Okay, who else? Because you had something true in what you said.
I think the structure argument was...
Yeah, exactly, because you were very close to the solution, is why. But can you pick it up, Andreas?
Yes, because there seem to be regions on the left side where the structures where partly are the same, and then it's a little break or something, and that then continues to be the same again. And on the right side, it's just chaotic.
Exactly. So the simplest way of looking at that is when you look at multiple sequence alignments, and again, what I mean here is there's the protein running the first residue to the last residue here, and there's another protein that is aligned, so the similarity extends over where the line is drawn, right? So the bunch here, a bunch here, and we have already discussed that this is the kind of thing where you say, well, there's something about the domain going on. That's one way of looking at it. Another way of looking at it is, this is a region where you will in fact have longer insertions. So this immediately, observing this kind of thing, immediately tells you that you will have more likely something like this than something like that. But let me go a level deeper. How do you imagine? What's the reality of a gap? So again, we're looking at evolutionary relation. We're looking at similarities in structure. So what do you believe? How could you imagine a gap? Picturing a structure, how could you imagine a gap?
Is it a missing domain?
A missing domain, that is essentially the level, I mean, behind, down here. Something like that is slightly different, where we would have a different, one domain would not be there, or something like that. But more on the level of actually lengths of this, so 5 residues typically is not a missing domain. So take the alignment or the number that I show here seriously, and tell me what sort of interpretation of a gap that has to do with the word structure would make this more likely than that. So how do you imagine a gap? What's the molecular explanation of a gap? Or so there's this one protein that looks like this. And if you looked at the same protein with a gap, what would the gap be?
Maybe a loop or something.
Exactly. So the same protein here with a gap may be one that does this. And in order to form a loop, in fact, you need, and let's talk about the happiness of a protein now. So proteins are not happy if I want to force them into one residue loop. In fact, it's impossible. You break the chain. So in order to form what people call a hairpin, you need at least 2, typically more 3 residues. So this is why that is very unlikely. The underlying idea being that you have, in fact, and this is also the 3D comparison story, you have embellishments, you have additions. So you can somehow, there's something that this structure can do, and by putting something else on top, maybe it's a new binding site. Maybe it buys you a little bit more flexibility. Maybe, I don't know what, and maybe it doesn't hurt. So there's a variety of reasons. But in the way we really mention them, there's something on top. Okay? And something on top is much more likely in this model than in this. Now, this brings us to the next issue. So if that is my objective function showing the same thing here, how can I enforce that we see more the left side of the story than the right side?
Can introduce gap opening penalties and penalties for extension. So.
Yeah. No, wait a minute. No, that doesn't work. So the gap residues, yes. No, now we have to have it. We have a gap open times the numbers of gaps minus gap elongation times the number of residues in gaps or length of gaps or something like that. Exactly. Now, all we need to do is have a smaller value for the gap elongation than of the gap open, and we immediately enforce that this is much more likely than that. Typically, in fact, this is differing by roughly a factor of 10. So the gap open is typically a factor of 10 smaller. Let's not get into that. And let's get into this one here. So far what we looked at is, on the score here is the number of matches minus gap. Is that the best criterion? Oh, this is still on, we are still on the side of global, but you know that the global alignment is not the right solution. Let's get into a different question first. In this particular example that I show, so you could align GQ against GQ and say this is my alignment. Or you could extend the alignment until here and would then essentially have this alignment. Again, yes, I should choose a different example with 2 gaps together somehow. But my question here is, what is better, 2 and 2 right or 4 and 7 right? Okay, let's turn that around. I assume that some people in the room are tired and some people in the room just don't know the answer. And that's why nobody says anything. The ones who do not say anything because they don't know, what question would you ask me in order to decide?
What you want to do with the information you got there, because if you have only 2 of 2, then you miss the information of the rest of the chain.
I want to predict that 2 proteins have a similar structure.
The 4th one is way more important because like the probability of its actually matching 4 is lower than probability of 2 matching 2, right? So basically 1 in 400, that would be like, I don't know, 1 in 220 to the 4.
Is that true? Is the probability of matching 2 and 2 higher than the probability of 4 and 7?
Yeah, it's higher actually.
Everybody happy with that? Think about it. We do not know the answer to that question. But the point is, this is the question you have to answer in order to answer my question. You have to, in fact, exactly compute the background probability. And seeing the background probability, one important question that you have to ask, if that is your match, how likely could that have arisen by random, which is exactly the background probability. So you have to answer that question. It may or may not be true what you say, but that is exactly what you have to check out in order to answer that question. The next thing you would have to answer in the context of predicting similarity in structure is, on the level of similarity of structure, what you essentially get out of this first step is statistically significant. The second step then would be, is that scientifically significant? So is there a point if, in fact, it turned out that this is not trivial. In this particular case, you are right. But assume this would be 10 residues long, and you would have 10 and 10, then the next question would be, if you have 10 residues, do they always imply the structure? Or could you, in two completely different structures, have the same identical 10 residues? So that would be the scientific question. The first one was statistic, and the second scientific. And you have to always begin with the statistic because that's simple to do. Now the next question is the question of, is identity, so far what I looked at is amino acid identity, is that the best substitution matrix? Is that the best way of compiling the match? And you're already well enough into the game to know that the answer is of course no. We have here an example of a potassium channel where in fact this alignment is extremely important. This is sort of the base to a Nobel Prize, this alignment, to figure out that in fact this is the part that senses the potassium in this channel, and through that alignment McKinnon could in fact solve the structure because they simply could use a bacterium that was so far away that from sequence comparison it was not recognizable, but had a very good intuition that the residues that were replaced here in the filter were biophysically tolerable. So clearly what we learned from that is we should have a scoring matrix that is not identity. And now comes history. We talk about PAM. PAM has absolutely no reality today. I don't know, maybe there are 50% of all the publications that use PAM, but it's completely outdated. It's not, it's in the space of people talking about the principle. PAM is very important. But in the space of people really aligning proteins, it has no relevance anymore. But historically, so this is Margaret Dayhoff, and she simply had about 1,500 different sequences, proteins in 1978, that she simply all aligned. And what she wanted to do by this alignment, she aligned with her eyes, in fact, sort of postulating that she has a feeling for alignment. She was enough of an expert to do that, and by all of these alignments, then she could go and simply count how much variation do you have. And she translated the variation into building a tree. In fact, this is the first time that the idea of a tree within the world of proteins evolved, or genes. And it's, in many ways, the Dayhoff's work really set the pace and changed the world. This one, by the way, was still also published in a book, where PAM comes with numbers, PAM-N, and PAM-1 is 1 point mutation per 100 comparisons, and PAM-N is the exponent of this. But again, historically interesting. You find a lot of web presence for that simply because it's so simple, not because it has any contemporary value. What has a lot of contemporary value, however, is BLOSUM. Stephen Hanikoff, because I couldn't find a photo of Roja. Stephen and Roja together published this paper on the way to build substitution matrices from log odds. How do you start this? How do you get, so in the case of Dayhoff, she had 1,000 proteins that she manually aligned. So in order to compile log odds, you have to have a set of proteins that you know are similar. Right? How do you do that?
If you take proteins of organisms of which you know that they are evolutionarily close.
So you take mouse and chimpanzee or something like that, and then you simply look at all the corresponding proteins, and for all of those you look at the changes. And in some sense this is, the BLOSUM matrices come with numbers. 62 is what I'm going to show you on the next slide, where n essentially is the cutoff in sequence identity for what you consider. So essentially you build alignments here optimizing the objective function sequence identity. That's your first step. And it could be even higher value, 98, so where you're sure that the proteins are really, really related. And then you simply ask how many vary and assume that all the variations are neutral, but hence this gives you a substitution through the log computation, a substitution matrix that reflects the biophysical relations. It's a little bit circular, but this it works very well. So let me just point out some of the facts that are all clear to you. The first one, and sorry for the bad quality of this slide. The first one is now we have on the diagonal values that differ. So matching the letter, not all letters are equally important to match. Is the first statement. So the tryptophan down here is much more important to match than in this particular case the alanine here. And the next statement that is important is sometimes you have off-diagonal matches, so matching a leucine and the methionine. I thought this would be an isoleucine. There's something. Okay, I owe you another one. There's something wrong here. So some of these matches off diagonal from hydrophobic to hydrophobic are in fact more relevant than matching alanine on alanine. So these are two aspects. The other aspect is there are a lot of negative values in here. So in the context of what I described so far, the percentage sequence identity, you only have 1 or 0. Here you have negative scores. And in fact it turns out that the 0 level, so that the overall value of this matrix should give you a slightly negative value for random comparisons to do the best or the fastest way of alignment. There are many substitution matrices available. Ultimately, most people keep using the BLOSUM 62, and let's just keep it there. There's a nice tool from Francisco Melo that allows you to play around with sequence alignment methods. Dynamic programming is optimal, but so there are two issues. One is related to time. So time is essentially proportional to the length of the tool. So assume we have L square. Now L square, typical protein is how long? Just take a run of the mill. There are many longer and many shorter. What is a run of the mill protein? 100. Typical run. This is in fact exactly what a domain is. Most proteins are multi-domain proteins, so it would be more than that. So run of the mill is more. Again, the average doesn't really make sense in this, but if this is more like 200, 300, 400. Okay, any number. 100 is too short for an average. Again, there are many proteins that are that short. Or many toxins, almost all toxins are that short. So there's a lot of important function, but still, most enzymes are sort of more in the 400 ballpark. Anyway, 400 against 400. Is that a big number? No. Somebody said no, somebody says yes. What's the distinction between these two groups? Optimist and pessimist? Or if I asked you, so I give you a dynamic programming to run on a phone, and I want you to do that before you leave this room. So I'm not going to keep you for ages in this room. So before the lecture is over, do you believe that the job will be done on your phone? Easily. Absolutely easily. If that is so easy, then why do I have a problem?
You do one pairwise alignment.
Exactly. That's where we get into it. I wanted to illustrate it in a even slightly different context here. When we came to Germany, our group, so we are running servers that in fact do alignment against everything. Break protein is the underlying server. And those days we had 8 million proteins in the database. In 2014 we had 50 million roughly. This is sort of double every 2 years. So this increase is just like the increase of computers. Today it's a little bit faster. Computers no longer do 18 months, no longer actually do 2 years, but similar ballpark, not that scary. But the real important issue is the big ballpark issue is that in order to do the comparisons here, you had 64 by the 10 by the 12, and here you have 3 by the 15. So that now translates to 20-fold increase every year. And that is way beyond what a computer can do. In terms of storage, the number is not as dramatic. It's only 5-fold because many of those will not lead to a hit, so you can ignore them. But actually in technical ways, so we came from New York at some point with 5 terabytes. Today's 300, more than that. When I did the slide, it was 300 roundabout. And the point is, when we came, I carried disks that had 2 terabytes, and it was fairly easy. But this one here, now many years later, you don't have easily carryable disk with 150 terabyte. And not only that, to in fact move the data around is complicated. Many of you may at home have these 4 terabyte disks for backup of their local machines. But if you lose one of those and do an entire backup, how long does it take? Any idea? Have you done it recently? So 4 terabyte is already days. Depends on your data, depends on all kinds of things. But on this level here, even big machines have serious issues with this. Okay. Now, another issue here is how to choose parameters. But for instance, gap open penalty, gap elongation penalty, other penalties. But the more important issue really is, how can we speed up? The more important question is, how can we speed up? How can we?
Try to parallelize it, but
Yes, you can. But then you have, that buys you all kinds of other problems and not really a solution. Because if you think about it, so this is the one of the many problems where parallelization doesn't help you at all. Because essentially, you can just run, if you have 5 processors, you can just run 5 jobs. In particular, if you could run an entire database. So this is totally parallel, right? The problem. And speed you don't gain. I do not remember what the dynamic programming against an entire switchboard would be. But I do know that the version that speeds it up, that is, oh God, I do not remember how much faster BLAST is, but at least 100 times faster. Now that process takes 1 hour for one sequence against the database. So you do the times 100, and then you get you quickly convince yourself this is not trivial for even the LRZ machine. Because before that is done, you have the double database and you have to, whatever. How can you speed up? You all know the answer. What's the idea behind the method that you all have heard of that answers that question in the simplest way? Well, not in the simplest way, but the one that did it in the most lasting way. Hashing, right? So the BLAST does it fast matching words. So you look at words, and words here are BLOSUM matrix type of words. So for the triplet that I show, you look up in the BLOSUM matrix whether the sum over the 3's in the BLOSUM matrix higher than something, right? It's not identical words. But think about it as matching identical words, right? And you can essentially put the entire database, we can split the entire database into these triplets, put that into a tree structure, and then now you come with your new method, you search against that, and it's very, very fast to find all the possible 3 mirror matches, triplet matches, right? But now the question becomes, what do you do next? The sequence alignments are not 3 residues long. In fact, sequence alignments somehow should, in particular in the context of this lecture, what we try to capture are domain similarities. So domains are about 100 residues long, so maybe we do half of it, but we want to do 50, not 3. So what's the next step?
You extend the alignment on each side of the C.
Yes. How do you do that? There's somebody else. Let's forget the word BLAST here. How could you do it? I'm not asking how BLAST is doing. How could you? So yes. So next step, you have this word here, EKA, and now you want to extend in both directions. So let's just begin to move in that direction here. What's your next question? How could you do it? It's very simple, yes?
Look whether the next pair of amino acids will increase your score or whether it doesn't.
Actually, the solution seems to be, there seems to be no other solution, but actually even in what you said, it's not that simple. You already made an assumption, Gregor. The assumption that you said is, okay, I'm going to go one at a time. This is the first assumption. I'm not going to go the next three. I'm going to go one at a time. The second assumption you made is your criterion for acceptance or removal is whether the overall score has an influence. So you could now make your decision on, is the next one going to match? Forget about the overall score you have and just ask, is the next one increasing the match score? Or you could say, my overall score goes down, or the average score goes down. Right? Which in fact, the way you propose it is the way the first BLAST did it, where you're allowed to, in fact, also align things that are not related, hoping that then one further you get to one that is related again. And you extend as long as you're not doing damage in some sense. While in the strictest way, you would just go one at a time and say, if that matches, yes or no, stop. No matter what I got so far. Right? And that you do in both directions. Now, this comes again at the slide. But do you happen to know how, I'm not even having a quote for BLAST. This is the Essential 93 BLAST, published in 1993 by Chul et al. How is the more modern way, published in 1997, changing on the pairwise alignment? It's called, that was introduced as with the word PsyBLAST, and we will get PsyBLAST. We will get back to PsyBLAST in a minute. And the most important concept of PsyBLAST is multiple and repetition and blah blah blah. This is not what I mean. There's another idea that is new in this 97 paper that did this slightly different. And that idea was in between. We're going to use dynamic programming. Okay, we're talking about speed up. So how can you do speed up if you go back to dynamic programming? Anybody?
Maybe you get a better idea of what is a relevant match and then have to do fewer local alignments in the next step.
That is already true. So you have to do fewer local, but why is it so important? What's the, you have 1, 2, 3, 4, 5 words, and that already breaks up my problem. So instead of one long problem, I have 4 small problems. But why is that so important? Let's just assume we had one word match roughly at around about half the sequence. Why is the gain not just half?
Because a quarter of the original.
It's the quarter. Exactly. So you're suddenly down, by finding one hit in half, you're suddenly quartering the time. And already you see with these many ones here, you break down the problem to much, much simpler tasks. Because it goes square in the length. That's the reason why after this initial extension you can easily apply the dynamic programming if you do the first mapping right. Nevertheless, dynamic programming, whatever you use, there will be regions in between. What about those? What does this method BLAST say about those regions? Nothing. The method says as much as you all do. As a common group, absolutely no statement. This cannot be aligned. I cannot align it by some thresholds that are given in the process. Word length, minimal match distance, how far can the overall score fall below something. By all of these criteria, I do not find them to match. So BLAST will report in this particular case 1, 2, 3 alignments at different positions. So will be with the first, second, and third. And they will all come with a different expectation value. Just to bring faces back, again, science is communication, is networking, and it's important to remember who are the people behind. FastA is a similar idea as BLAST. The major difference of FastA and BLAST I'll get back to in a moment. Bill Pearson, and that is in fact from Bill Pearson it comes. The person who is on both papers here is David Lippmann, NCBI director for many years. And then there's BLAST, Stephen Altschul is here. And other people, Eugene Myers and Webb Miller, who were on the initial BLAST papers, I would argue, David Lippmann is here again, that the main person behind BLAST really is Stephen Altschul. Webb Miller went ahead and actually did ancient sequencing of the mammoth, for instance. That's the project that he started. Eugene Meyer was one of the people who assembled the human genome and now works on understanding neuroscience better. Why do I bring that? Not entirely sure. The major issue that we have in BLAST is matching, or is compiling expectation values. So as long as we have the objective function of dynamic programming, we're in a sort of okay world. But when we have short matches, we have to establish statistics. How do we compile this expectation value? Not in detail how you do it, but what's the idea behind the expectation value? And this brings us back to the question, exactly your answer that we had, is 2 and 2 better than 4 and 7? You essentially look at the score versus distribution. So what I mean is you have some score for the hit that you actually observe, and then you have some background. And the E-value is proportional to the difference between the score and the major part of the distribution. The difference is influenced by the average, by the standard deviation, and by the height. Now, let's get back to the height story here. Say that is my alignment, okay? And there's a certain E-value for that. What does it mean that the E-value depends on the height of the blue? That means that if I got that match from running against a small database, I get a different E-value than from running against a big database. Is that right? So you run BLAST and find that alignment running against SwissProt, you get a different E-value than getting the same, with the same settings and the same BLAST running against UniProt. Is that right? Something that you have to remember. Yes, it's absolutely right. And can you rationalize it?
We have different background distributions.
Yes.
For databases.
No, but be careful. Oh, yes, no, good point, Jade. But I assume, okay, let's turn that differently. Let me make another experiment. I take SwissProt and I make 10 copies of SwissProt. So now I no longer have different distributions. Right? I have exactly that distribution except for it's 10 times higher. I'm not changing the composition. You're totally right in what you say, and I made a mistake. This is an important point. I should have made that more clear. So UniProt, SwissProt, you're right on. I believe this is a very important effect. But here, what I really mean about the height, there's another effect. And that is also very important, and that sort of often is less intuitive, which is in fact that by merely increasing the height, you change the E-value. Why?
Because you have more sequence in your database, right? I mean, like if you have a sequence in your database, the probability of having it, if you have 10 folds of similar, you will have 10, right? Instead of 1.
Yes, but you have to essentially how can my E-value change by just making 10 replicas of the same thing? I'm not doing any other search.
It's basically multiple testing, right?
That's a good one. You shouldn't actually do it, you say.
No, you do a Bonferroni correction, and that's why the E-value changes.
Does BLAST do that? No, okay. No, of course it doesn't do that, because nobody's ever going to be so mean to put the same thing in the database again. But remember, there's a high redundancy in UniProt and SwissProt. And BLAST does not correct for that, and cannot correct for that, and part of the reason why it doesn't and cannot, I will get back to in a minute. But let's look at this example again. In fact, that's what many people do. They take proteins that are somehow related from different databases. Two different databases interact that have protein interactions. You just put them both together, and then you would get an effect, and you would run a line against those data, this new database, and you would not make multiple testing correction, because you simply run BLAST. You convert your database into a binary, and you run BLAST, and you get two different e-values. Is that right? For the example I make. It is right, this will happen, but it's actually fake. I mean, it's wrong. It's scientifically wrong. Technically, there's a reason for that, and the reason for that is, essentially, you have all said that. If you say you want to find out whether the letter, the word art is a good way to come up with a keyword to classify publications that have to do with math, and publications that have to do with art. And I'm going to give you 5 papers on painting and 5 papers on chaos theory. And then I'm simply going to count. And in those 5 papers, if you had 5 papers only in this painting, we would have a 5-0 occurrence, then you would have a certain trust that this is a good keyword. But if you did the same thing on 500 papers, your trust in the right choice of a keyword would increase a lot, only by the number. Again, so my example of repetition is misleading in this sense. But even if you had still, if they all would be a sort of a similar, sampling from a similar universe, but just the fact that you have seen more, your trust, or let's go to the other extreme, you have seen only one. You can always find ones that in a math paper on chaos, and you find art, and a painting paper you don't find it. So you can always find exceptions, right? This is why small numbers clearly, so that's the rationalization why this is the case. Underneath, the math is very simple. Now, what is really different here is the big difference, in fact, between FAST-A and BLAST that is earlier, and that survived because this one is much substantially 10 times slower or something like that. The big difference is that FAST-A, in fact, takes the sequences you run against and does a complete permutation and compiles the actual background probability, which is absolutely the right way of doing it. BLAST, do you know how BLAST does it? It's already written. It's pre-compiled for the database. Essentially, all it ever uses is the composition. So it simulates the distribution based on the composition. And that's why it can be so tremendously fast. And this, of course, is much more dirty than that, but it's so much faster that the level of dirt or the lack of performance somehow was accepted. In fact, it turns out that I personally believe that FAST-A didn't survive because it is just the BLAST survived because having the breakdown into words and then couple it to dynamic programming is so much winning. So you do it so fast and so accurate by combining the best of both worlds, which is getting more speed and more performance than the correct solution in between the two. So 2 hacks are faster and more successful than one correct solution in this particular case. It's not always like that in life. But this is an example where I believe this is my explanation why it happened. Certainly it did happen. How accurate are pairwise alignments? And let's begin with a bunch of questions. Similar sequences, do they map to similar functions? Similar sequences to similar structure? And assume that we could map, compute, draw the universe of all sequences. Again, we don't know it now, and of this we know much less than of the universe of all structures. But say this is the universe of all sequences, and this is all the universe of all structures, so some sequences map to some structures, as I show, and similar sequences map to essentially the same cluster. To similar structures is the right answer, but I call it one dot because you have a 3D comparison method that would put them onto the same dot. Okay, that's the idea here. And then there's this picture here. Most sequences don't fold at all. Why? So again, this is an assumption at this point. You don't know most sequences. But why could that be the case? All possible sequences.
All possible sequences we can find, or you can imagine.
Yeah, that's exactly the difference.
So some sequences.
All possible sequences we can find, no one like that. So if the sequence space here would be the real observed sequences, we wouldn't have any of these.
Yes.
Okay, there may be some it's biology, there will be some. I cannot really understand why, but I bet you there will be some there. And maybe we will discover what they do. But what I mean here is by the universe of all possible sequences.
How would you define fold? I mean, like if you just fold, like it moves, it kind of folds, but it's not really folding.
No, so one way of defining fold, so again, in operational definition I have two things that I can compare and I can cluster. The operational definition, however, of fold is you take the sequence, you throw it into the solver, it adopts a unique three-dimensional structure, you do it again at a different time point and you get the same thing. That would be the definition of fold. And in structural determination, essentially the fact is you get something which you can shine light upon and it doesn't wobble all the time. Two different time points you don't get the different solutions. Anyway, now is there any question at this point?
Is the statement true? Because I'm not really sure it's true.
That many sequences don't fold. Which part about the most? The letter word most? I do believe the statement is true.
How? What's the explanation?
I do believe it's not that. So now you get into something that is much more involved. But ultimately, the explanation is you do not. So the sequences we observe is the set of all the sequences we know today has very different statistical features from the set of all sequences we can imagine. And why is that the case? Most likely because you have correlations. You have to have certain dipeptide formations, and that means that not all pairings are possible to make structures of. And that then would explain this. So if you pack together, and this is in fact also one way in which you find non-coding RNA, by creating things that are totally hydrophobic, or just have iterations of hydrophobicity and not hydrophobicity that are not observed. So I'm going to go a little bit faster in the slides here. So there's a daylight zone, so there's a realm. I want to predict that from sequence comparisons, I want to see whether two processes have a similar structure. There's a region of sequence identity, and these numbers are just placeholders, give rough ideas, roughly right, where the comparison is very trivial. Two processes have 100% sequence identity, they have a similar structure. 60 there's still, and then there's a twilight zone where the signal becomes, and that term comes from Rost Doolittle. I showed the photo on the slide before. The twilight zone where we sort of lose the signal. So sometimes still proteins pairs in that zone still have a similar structure, but sometimes we make mistakes. And then there's a midnight zone where they really don't have similar sequence enough in order to further have similar structures. When we get into 35 to 15%, again this is placeholders, fake data. All I want to show you is what we sort of expect. We expect that at some point we can infer the similarity in structure from the sequence comparison directly, and I was right. And then, going to the twilight zone, we'll see that the signal will disappear. But at the same time, not looking at the ratio, the accuracy, but looking at the raw numbers, at this point, the red of false, things that do not have the same 3D structure, comes much closer to the blue, but the blue rises. So I want to get into the twilight zone to get all the matches. Okay? Somehow we expect something like that. How can we assess alignment accuracy? Usually this is the moment where we form groups and discuss it. I'm trying to get through the rest of the lecture. So what you have to do is you have to take proteins for which you know the structure. You do a 3D comparison, a 3D alignment. And then you have to do something with a sequence unique subset here. Let's not get into that. We have proteins for which we know they are similar. We have proteins for which we know they are not similar. And we have something in between that we will ignore to make a clean data set. We do an all against all. You have to do a sequence unique subset, and then you have to align the sequence unique subsets against all, and then you build a curve that initially was done by Chris Sander and Reinhard Schneider. This curve here, 100% sequence identity to 0% sequence identity. Sorry, here's the percentage sequence identity, here's the number of residues aligned. And then there's a saturation point. Let's not discuss too much on the curve because the curve changed over time, but the idea of the curve is the same. So again, the reds are pairs of proteins that do not have the same 3D structure, blue pairs of proteins that do have the same 3D structure. That's the curve I showed you before with the slide changes. And you see, it is not true that there is no, the curve is not such that there is no red point above. But somehow, and the curve is also not such that there's no blue below. But somehow it looks like a lot of the blue are above that curve. And if you move this curve a little bit more here, you would in fact essentially get all above that curve. What you don't see here is the density. The curve really is such that there's not much above it, although it looks different. Now, in terms of density, what we're going to show now is I'm going to look at distances from this curve. So this is the curve I show. And now I'm going to ask how many are right or wrong if I go +5, +10 and so forth, or -5. On a level of 100% sequence identity, well, maybe let me just make this one point about the curve. What the curve implies is if you have 100 residues aligned, and 33 of those are in fact identical, then essentially these two proteins have a similar structure. For alignments longer than 250, this is close to 20. And for alignments of 10 or 11, this is not even at 100, meaning that if you have 11 identical residues, back to the 2 and 2 and 4 and 7, 11 identical does not tell you anything about structure. Okay? That's one outcome of this curve here. And that's reality, because you can find the same 11 mer in different structures. But this is essentially the curve. Now we're going to look at how well do we infer similarity in structure from sequence comparisons with respect to that curve. The distance with respect to that curve. I put sequence identity here, and that is the value, I guess the value for, it's not 100 residues. I don't remember what the value is that I look up. But this is the data is really done on this axis. This is just for memory. Hi guys.
Hello.
Should we restart?
Yeah.
Okay, so the curve is actually the zero line, and what you see here is you, in fact, going into the twilight zone of the curve is logarithmic. You see that the pairs of proteins for which you have similar structures that you want to find is increasing tenfold, more than tenfold, by more than a factor of 20. So more than a factor of 20 means you have a high incentive to get in there. Now let's look at the faults. You go from something that is very close to 0 here to something that is very, very high. In fact, going from 8, or wherever the 0 was, a level of 8 to a point 0 to 10,000, to a point where you have equal numbers, 50-50, is only in the scale here 4 levels. So it's almost the same curve. You just move it 4 points on. And you move from no mistake to 50-50 mistake. To another roughly, where's the crossover point? Another 1, 2, 3, 4 points. Well, round about here, you are in more than 10%. So now what you find is less than 1 in 10, you're right. 9 in 10, you're wrong. And all of that happened from 0 to 90% mistakes happened within 8 percentage points. Essentially 8% sequence identity for this given alignment length, right? This is a remarkable increase. It's like an explosion, a phase explosion. So far, what we did was pairwise alignments. We can extend that a little bit. Can I still run through the last? Yes, easily, right? So far, what we did, free will. I'm a complete defender of free will. You have the freedom to stop me at any single time, at any time moment. Okay, back to this issue. What we so far have, a pairwise alignment means that you essentially have your query protein at the center of a circle. Defining a cutoff in the pairwise alignment, H is P distance, pairwise sequence identity, E value, whatever you take, you always essentially draw a circle around it. Okay? Now, assume that the situation is such that I have a protein A, a protein B, and a protein C that are all too far apart for the threshold that I want to tick. H is P value, H value of 0, E value of 10 minus 3. We cannot infer their similarity. But now let's assume that we have another protein X, and that protein X is within the safe zone here of A, and the same X is within the safe zone of B. What does that induce in you as a guess? How could you possibly use that?
Well, obviously the protein X has regions, like at the front regions are related to protein A.
Okay, let's assume, Andreas, let's assume that we have a full domain. So that what we're talking about really is on the level of an entire domain. What would you learn then from it? You have a very, very good point. So it could be that X is a two-domain protein. Would be a simple way of addressing that. Assume that the alignment of X with A is in the same region of the alignment between X and B. Sorry, it was complicated. I know it's late, and now life is tough. A, X, B is the alignment. What can you immediately say? Yeah. Exactly, that they are related, right? So you simply will infer that B and A are similar, have the same structure. And if you did that, remember I said that it levels around -2, or here around somewhere here -4, you already had 90% mistakes. Here you stay at 100%. Down further. And we get into the next level of multiple alignment. The first one, let's just do it quickly. You can do a three-way dynamic programming, and it's also sort of easy squeezy, even up to a level of 60. You only need 60 terabytes of main memory, which is totally easy. Is in principle doable. Practically, we have a machine here with which we could do that. I don't have a quote for this, but I've heard that in some conversations. But you begin to see this is not doable. So in order to do larger multiple sequence alignments, you need to find a hack. And this hack essentially comes from Rost again, the one from the Twilight Zone, in which you break. And that we will discuss on next Tuesday.